All right, we're going to go ahead and get started. Good afternoon, everyone. Welcome to today's Science Luncheon at the Center for Vital Longevity. It is my pleasure to introduce today's speaker, Dr. João Moreira, Assistant Professor of Psychology at the University of Wisconsin-Madison, where he leads the Computational Developmental Neuroscience Lab. Dr. Moreira's research explores how young people develop the cognitive, emotional, and social skills needed to navigate an increasingly complex world. His lab investigates topics such as emotion regulation, risky decision-making, and social cognition, often using cutting-edge methods like fMRI, ecological momentary assessment, and naturalistic text analysis. His work explores how our brains develop the capacity to understand and relate to others, particularly during critical periods of late childhood and adolescence. His research has been supported by the National Science Foundation and the National Institutes of Health, and he's received recognition from the American Psychological Association for his contributions to the field. Today, he'll present his talk entitled, How Do Daily Experiences Sculpt Mental Models of Others?, offering insights into how our everyday interactions shape how we perceive and understand others. Please join me in giving a warm welcome.
I'm thrilled to be here for you all. I've never been to Dallas, so it's nice to come in here and get to know the city a little bit and talk with folks at such a cool center. Now I'll note here for this talk, I'm going to share preliminary data of an ongoing study. So I'd love to get feedback on anything that you want to give feedback on. I'm excited to share this work for the very first time with someone else. I've talked about the study before, they've not talked about the findings. They are still preliminary, so just a heads up. And also this is the type of study where we can kind of make adjustments on the fly, as you'll see. So we have about like a third of the data in, and so I would love any feedback on the design. I'll start by telling you a little bit more about me and dive in. So I'm a developmentalist by training, so there's my obligatory baby photo. I was born in Brazil. I go by John or João, either one. I truly don't have a preference. I think people think, oh, you care one or the other. I don't. I like having a name that's multiple choice, João or John. I moved to the United States when I was really young, while y'all were doing your 90s thing. And that's why I speak English like this and Portuguese with an accent. I'm going back in November, and I'm going to surely get teased for my gringo accent. And growing up in a Brazilian household and navigating American culture outside the home made me really attuned to cultural differences in behavior and made me want to study psychology. It'd be really cute if I became a cultural anthropologist, but I didn't. I just thought why people behave was an interesting question. And so when I went to U of I for undergrad, see the line I shirt back there, I joined Eva Telzer's Developmental Social Neuroscience Lab because I actually hated developmental psych, but I like social psych. I like neuro. There were 2 social neuro labs on campus, and one wasn't taking RAs, and Eva's was. And by the end of that first year, I decided I want to stick with development because it's really cool. And I went ahead and got my PhD with Jennifer Silvers and her Social Affective Neuroscience Developmental Lab at UCLA. Loved LA so much, I stuck around to do a postdoc with Carolyn Parkinson in her Computational Social Neuroscience Lab. And now I'm at Wisconsin, where I teach this where I direct the Computational Development Science Lab. And the primary goal of my research program is to understand developmental mechanisms that allow individuals to adaptively navigate their internal and external worlds with specific emphasis on adolescence. And I think of their internal worlds as being how youths tend to regulate their emotions, and the external being how they make choices in a variety of contexts. This manifests in a 3-pronged research program, and I'm going to talk to you about this latest one here. This is the kind of newest prong of this work. So throughout our lives, humans are tasked with encoding, storing, and retrieving and using information about various individuals, whether it be ourselves, our closest friends, and family and colleagues, public figures, or even fictive individuals. Our brain has to have a mental representation or model of each of these people that stores information about them, updates that information, and uses it to guide behavior. Right? This is nothing groundbreaking, by the way. This is all it's not unique to social neuroscience. It's been, in the field since like the cognitive revolution in the 50s, maybe even before then. There's some tome I haven't read from back then, but this is a pretty basic premise, right? Now, a bunch of studies in social neuroscience, I care about how this applies to people, have looked at the neural machinery implicated in encoding and retrieving information about other people. Several studies in recent years have looked at how markers of social behavior like loneliness or social network position are embedded in this neural architecture. Indeed, this topic has received a lot of attention in social neuroscience over the years, but relatively little work has examined fine-grained mental representations of multiple known others. So let's break this down. A few things to unpack here. First, it's really rare that studies examining how we think about other people, foundational sort of neural architecture for thinking about others, has not looked at this with known others. That's relatively rare in the literature. And of the few studies that do include a known other, they rarely, if ever, examine more than one known other in the study. And they often don't differentiate between given others within a category, like comparing the mental representations of two close friends, or two family members, or your polyamorous two romantic partners, you know. Love that joke. Go over that. Texas. Neural representations also tend to be elicited in a coarse way in these studies. They may simply take an average of brain activity or completing a task that requires one to think about others without explicitly modeling the of these tasks. I'll talk more about that later. So this means all those papers I just showed you on the prior slide may not apply very well to the most relevant interactions of your life. Every one of you got up today and saw someone that was close to you. And you're seeing these people right now. You all work with each other or know each other. You have friends, you have family, you might have children, but none of those papers on that prior, I shouldn't say none of them, but a lot of those papers on that prior slide don't look at that. They look at a generic other or a fictive individual or a celebrity or what have you, right? So this is kind of a problem. Two, little work has quantified how experiences with others sculpt and resculpt these representations. Theoretically and practically, these mental models are sculpted and resculpted with our experiences with others. And I say practically, I think, because you don't need a scientist to tell you that. Most of you met me 10 minutes ago, right? And from when Kendra read her blurb about me to right now, you have instantiated a mental model of me and you have been updated. And that model is going to change if you were to ask me to go hang out. Like, would he like coffee or tea? Would he like to get whiskey or gin? Would he like to watch golf or football, right? And the feedback you get in response to that is going to update that model and is going to guide behavior in the future. So there is this reciprocal chain of events that happens. This has also not been examined in the literature, right? So that's what I mean by, well, I was going to say this by practical, it doesn't matter here, right? So this underscores that these models are fluid, but most studies treat them in this static fashion. And finally, little work has captured the dynamic nature between behavior and mental representation. So it's kind of related to the first bit, right? But that these models, these mental models are being updated, right? And they also drive behavior. So there is a link. It's not just that they are being updated with new information, they drive behavior, and behavior nudges them to be updated in a certain way, and then they continue to drive behavior, right? These two are kind of related here. And so some work has looked at how the sort of like psychological architecture of these models are related with behavior, my own included, but very few have looked at how this happens in a dynamic, sort of intensive way over time, right? Which leads me to the main question that motivates this line of work in the study, and the data that I'm going to show you are how are fluctuations in mental representations of close others and social experiences linked over time? And right now, the way I think about this goes back to a study that came out almost 10 years ago. If you don't know, this is a study by Alex Huth and Jack Gallen. It came out in Nature in 2016. In fact, I think it's one of the very few last single lab fMRI studies to be published in Nature that I'm aware of. And I saw this study at the summer school, my first year going to grad school. And I have thought about the study once a week, every week since then for almost 10 years, which just shows that I need to get a life. But if you're not familiar, a handful of subjects, I think it was like 6 to 8 subjects, listened to about 8 hours of the Moth Radio Hour podcast in the scanner. And if you don't know what the Moth Radio Hour is, it's a narrative podcast where people tell stories. So like they range from really heartbreaking, like Hassan Minaj has a story about how his prom date is like a white girl who didn't want to be with an Indian man, ranging to like more whimsical or funny stories, right? And they regressed sort of all the semantic content of those stories onto the bold signal and pulled out this lexical atlas, this generalized lexical atlas that people use to sort of parse semantically relevant content in real day life, right? I defy you to tell me. So when I first saw this, I immediately thought, well, if this is sort of this generalized atlas, this has to be modulated to accommodate how we think about other people, right? I defy you to tell me that it's not, right? That the way in which, no, theoretically, but it's what the data do, but the way in which we think about other people is probably going to be like the way in which we access information about a friend or your child or a partner is probably going to modulate this guy differently, right? And so my thought is that the structure, functional architecture of this atlas differs between the people in your life, right? So these are two close friends of mine. This was on my last day in LA, a photo I took with them right before I left. My atlas for Armando is probably different from mine for Audrey. And differences in that functional psychological architecture are probably going to be related to how I choose to spend time with them, the effort I put into my what I do with them, in the first place, right? And this probably changes with experience over time, right? So this is sort of the key intuition that I want to look at in this line of work, is to pull out individualized or personalized mental representations of others and examine how they change with time and experience, using this idea of a lexical atlas, right, as an instantiation or as a model for how we represent others. And there's a bunch of reasons why you'd want to pick sort of a semantic atlas instead of another framework, right? Semantic content is privileged in psychological theories about person knowledge. It encompasses other organizing heuristics, how we think about other people, like the big five or the big two, right? Theoretically, it's all nested in language, the relationship between those theories of how we organize or how human behavior and knowledge is organized is in theory, is all nested within this. And it's also relatively clear and interpretable, right? So I'm going to show data of a study that geared to is set up to investigate this. I'm going to show you a bunch of tiles in the study overview, and then we'll go over each one. So in this study, we're aiming to enroll 5 to 7 college students at UW and have them nominate 2 close friends they have made, deliberately sampling college students in second years, specifically because individuals in this stage of life are going through a really unique time. They have made novel friends. They're away from home for the first time in their lives, and they are making novel friends. And in this sort of second year of college, they have made connections that are stable, but not yet crystallized. They've met people that they would call a close friend, but they're not quite so immune to change, right? It's not like this is my wife that I've known, been married to for 40 years, and like we know each other like back of our hands, right? There's still room for fluctuation. That's not to say that those fluctuations don't happen in other relationships, but this is an ideal test population for this very first investigation, right? And we're going to have them nominate 2 close friends. I'll talk more about that in a second. We will elicit mental representations of each other during scans. And they're going to do 20 scans over 8 weeks, each person. And we have 2 subjects who did this over the summer. We have 3 enrolled this term. Hopefully we'll get the last 2 in the next term. At the same time, they will complete concurrent daily checklist tapping behavior with these 2 others. I'll tell you more about what we are getting. And the goal is to fit a technique known as voxelized encoding model to the fMRI data and to pull out an individualized semantic atlas for each person's representation of an other at each time point, and look at how fluctuations in that content tracks with daily behavior. So that's the goal. Well, let's go through each bit of the study, and then I'll give you the results. What I have. I have not gotten to this last tile. This is a lot of data, as you would imagine, and so being very careful in how we analyze it off the bat, and trying to be really methodical. So I won't have the kind of end product is still in progress, but I will have some cool findings to share. All right, so let's talk about the two friends that they're nominating. All right, so we use a procedure by Rakelin and Jones. If you don't know about it or you don't want to read the text on the screen, we basically had our subjects think of a number line from 1 to 50, and their closest, friend at UW would sit at 1, and a very distant person would sit at 50. It might be a person who they see around a little bit and chat with, but they might not even know their name, right? So we had them nominate their closest friend at Madison that they made since coming to Madison. The idea being we're going to try to remove any between-person confound in sort of experience prior to Madison, right? Like, I've known my best friend for 20 years. You might have known your best friend for 5 years, right? So we want to kind of get rid of that. And also get friends they made at Madison. It's still a sort of like ideal test population, because I think these mental models are like the default variable, right? And so we pick their closest friend. And if you say, hey, pick your closest friend that's at like number 1 on the scale, so the closest to you on this number line, and then ordinally pick someone 3 or 4 slots down, but that you'd still call a close friend, right? Pick 2 close friends, so they kind of serve as a natural control condition to each other, right? To see if these effects are specific to, a given close other. And they couldn't be a family member, and they couldn't be a current romantic partner. And they have to be able to spend time with them voluntarily. These things are usually, this is the case when you do this. I've done this for other studies for almost 10 years now, and it's you usually don't have to specify these, but we did this with the small head, right? Then we elicited mental representations of each close other in the magnet. I'll tell you about the paradigms that we did that with. These 2 paradigms, it's an hour-long scan. We book an hour. It's about 45 to 48 minutes of scanning or so. It's 2 paradigms: trait judgment task and memory playback. What I call memory playback. The trait judgment task is borrowed from folks like Jason Mitchell, Diana Tamir, and Mark Thornton. That they have used this a lot. These are all folks in social neuroscience. So in an event-related manner, the subject rates preferences or traits about each close other one at a time in the scanner. So they'll see items like gets bored easily, is good about saving money, like goth night at the dance club, thinks art is boring, feels unsafe being in a major city, et cetera, et cetera. And it's not just a willy-nilly sample of items that we picked. So Jason Mitchell, who pioneered this task, had about 800 items on his website. Download those 800 items. I cut about 150 or 200 of them just because they're very old. They had stuff about like the Iraq War and do you think LGBT folks should serve in the military? At the time I optimized these items, that was not, we thought we'd settled that as a culture, but I guess not anymore. So we kind of like dropped a couple of the items that just were a little dated. And what we did is then we had them optimized to load onto the big 5. Specifically, we binarized the big 5 because each dimension of the big 5 is bipolar, right? So we chopped it in half, quantized it in these 10 bins, and then optimized it so each item loads onto 1 and only 1 bin, right? So from those like 600 items, we got it down to 130, and then they are shown a random subset of these items at each scan. And I'll talk more about this task than the next task, and I'll tell you specifically if how the stimuli are shown across the scan sessions. Right. But the idea is that these items tap the big 5. Again, the logic here is that the big 5 is a good descriptor of human behavior, human traits, and is probably a good way to tap how people think about others as well. And some of the task specs. Okay. Memory playback task. This other task involves completing a memory playback via a rapid serial visual presentation paradigm. So what this is you're just you read the task is where you read a chunk of text one word at a time. 1 word at a time. That's the the core gist of the task, what we did is prior to enrolling in the scan or in the study, our subjects wrote or they answered 30 prompts about each close other. And half the prompts tapped a specific memory they had with each close other, and the other half asked them to forecast something about the close other. Right, like where they're going to be in 10 years, or what's something you could see them struggling with, or what's something you could see them excelling at, or what have you. And there were about 4 to 6 sentences for each prompt, and then we show them a subset of prompts during the scan. So they read back one word at a time, the whole memory. The last sentence in the memory has a period at the end of it, and will not advance unless the subject presses their pointer finger to ensure they're still paying attention. And then there's like a 10 to 15 second break in between memory blocks. It's kind of like this event relate. It's kind of just like a block paradigm that is compatible with what other folks have used in this, with the kind of method, with the analytic methods that I hope to use. And we optimize these topics to ensure that they capture social information. So in a separate sample, undergrads come in and they wrote about this. They did these topics, and we just were able to use word embeddings or from the memories and try to predict features like relationship quality or closeness and similarity, et cetera, just to make sure that this is that variance across the memories and how they're written for a given prompt scales with meaningful social information. So I said it's 30 memories total, and there are 10 topics. So there are 3 prompts. I should stop saying memories. It's not exclusive memories. But there are 30 prompts, and each prompt is nested under one topic. So each topic has 3 prompts, and during the scan, they read back a response from every topic, right? So there's 3 prompts, so each of the 3 kind of little chunks of text per topic, and they will get something back. They read something back once for every topic during the scan. So we randomly pick one prompt from the first topic, one prompt from the second topic, and so on. And you can see here what these topics were. And the idea with doing this, and we had a similar thing with the trait judgment task, where they had to, we basically showed 9 stimuli per trait poll per other during the task. And the reason for this is that we wanted to kind of, the sort of organizing framework for how we're trying to sample representation space is consistent from session to session, but the stimuli vary slightly. So given that it's sort of a random draw every scan, there's going to be some overlap from session to session. Maybe, 2 out of the 90 items are the same, or like 12 or 15 out of the 90 items are the same in scan 1 compared to scan 2, and then scan 2 and 3 have, say, 14 items that are the same, or so on. And the idea was that we wanted to balance, sort of thread the needle between habituation effects and novelty effects. Right, so you can show someone the same stimulus over and over 20 times in the scanner, and they're going to get bored and they go on autopilot, and they're not, right? So our changes that you're seeing with experience are over time a result of habituation. Ultimately, you could change the stimuli every time, but you run into the same. So I said, let's sort of, if we think about these sort of 10 topics or these 10 trait polls as the kind of the sandbox of representational space that we want to sample, let's keep them in let's make sure that we sample from the same sort of quadrants of the sandbox, but maybe not the exact same sort of grains or spots. So that was the logic that we were using. And I talked to other, I spent like a year just like Zooming people who I thought would have interesting things to say about this. And it seems to, they're like, oh, it seems good, go for it. So, and this is the sample memory. This is one that I wrote, so I self-pilot, not any subjects data. But I piloted this on myself to talk about. You can see this is what memories. Most of them look something like this. So you would read like, I could see one of 2 things. This word would say I would need to press pointer finger. First, I could see him being blah, 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 blah. And then at the end of the memory, there's like a 10-second pause. So they did these scan sessions 20 times over an 8-week period. Admittedly, this is arbitrary, this number, both 20 and 8. It's been shaped by existing literature on intensive longitudinal data, funding constraints, and also practical methods. Could I get subjects to come in for 30 if I had the money? Should I get them to come in for 10? What I wanted to do is a lot of intensive longitudinal studies. I don't think have gone more than 10 sessions. And they've certainly never gone in a short time frame, which I think makes sense for the questions that I want to ask, right? That our lives unfold and we have meaningful events that can unfold in a 1, 2, 4, 8, 10, 12 week period, right? So we just kind of wanted to do something that would be that hadn't been done before. So in other words, try to push the sort of like boundaries of the sort of like temporal resolution. It's not the scanning parameters, but how often we sample. And so this is what the sampling scheme would look like ideally. So this is the idealized scheme where we're asking, no one's coming in on weekends to scan, and we're going to scan people, 3 scans 1 week, 2 scans the other for 8 weeks, and so on. And this is what I put in my grant application, this very figure here. And we're like, we'll have overflow sampling in case of reschedules, or we'll just schedule, 26 sessions, and then we can cancel those 6 if they come in, or for all 20. In reality, it was both messier but cleaner than I was hoping it would. So the scans were not as nicely spaced as you could probably imagine. But almost immediately was not feasible based on the schedules of our subjects over the summer when they have more free time. More challenging during the school year. So what that meant is that the minimum lag in between scans was 1 day, so you might scan on a Thursday and then again on a Friday, and the maximum lag was 5. So you might scan on June 10th and not scan again until June 15th. But modally, it was 2 or 3 days in between scan sessions. So you might scan on the 2nd and then scan on the 4th again, or could scan on the 17th and the 20th. Most of them had that 2 or 3 day lag. Compliance was really high. No one kind of bailed or canceled over the summer, and then I can tell you offline that we haven't had nearly as good luck here in starting the school year. But we were shocked. In fact, one subject got 21 scans. We had to get 21 sessions because the scanner lost 8 went up. So this was really good, and I was like very happy. And as a result, we didn't need this guy, which is really great. So we have complete data for everyone, all 2 subjects thus far. At the same time, they did these daily checklists that tapped behaviors of each close other. Now, checklists were sent out every morning during the intensive longitudinal period of this study. And to be clear, these were daily checklists. The scans are not daily. The checklists are. And they're sent out every morning, and the subjects were asked to complete them as soon as possible. And we did that because, we said as soon as possible, because we asked them about the prior days of experiences. These are college undergrads. I do not trust them to answer this before they go to bed. They're going to bed at 2 a.m. right after coming home from a party. And we don't want to say do it by 10 p.m. because you might hang out with your close friend after 10 p.m. Something fun might happen. I want to know about for the sake of the study, right? So we just said do it the next day and please answer about the prior days of experiences. And this was communicated to subjects verbally. It's in bold on in the text of the items. And we asked a variety of questions, and the data and the findings I'm going to show you in just a little bit kind of tap 3 variables, right? We asked more than this, but these are the 3 ones, and I'll tell you everything we asked. But first we asked, how much time did you spend with someone on this day, right? So these are daily, just in terms of minutes you spent with them. Then we asked questions, 5 questions, what I'm calling friendship cohesion. And when we planned this, we just kind of wanted to tap meaningful aspects of daily life with each friend. And I've aggregated them in a composite here that I call cohesion, but I'm not wedded to the So they are, did you support your friend? They help you. How emotionally close did you feel with them? How disconnected or lonely in your relationship did you feel with them? And did you engage in any kind of meaningful self-disclosure? So this is not just random chit chat. This is sharing feelings, struggles, big decisions that you're facing, hopes, etc. We also asked about susceptibility to peer influence. These were 4 items that were, did you do something with your friend you didn't want to, but you did anyway? Did your friend's opinions influence your behavior? Did you change your behavior to fit into what your friend was doing? Did you feel pressured to go along with what they were doing? And I care about peer influence here for a few reasons. First, it's an interesting test case in motivational behavior because you are overriding one goal that you have in sort of in response or to favor another. You may not want to, smoke pot with your friend, but you may want to fit in. So you have to sort of inhibit your, I really don't want to get high tonight, but I don't want to upset my friend, or I really want to sort of like keep being friends with the person, so I will go along with this, right? So it is to me an interesting test case in motivational behavior. You just think of it as like decision making in a way. It's also relevant to kind of like a long-standing history and social psychology about conformity and compliance. And it's also highly consequential to adolescent well-being. So susceptibility to peer influence is a huge predictor of substance use in teenagers. So for those three reasons, we tap this guy. And then we have a couple other things. In the interest of time, I won't tell you, but I'm happy to say, I'm happy to talk about all the items that we have measured in the EMA. It's just a couple more, and I didn't analyze them. I eyeballed them, but I didn't analyze them.
I may have missed this early on, but you told for sex subjects or different.
No, it's two women who did it. I thought about controlling I thought about sex matching the friends and the subjects, and I talked to some colleagues, and they were like, just do the people that are closest to them, right? So at the expense of sort of dynamics. But I think that's like an interesting sort of, we have some subjects that I think both our subjects over the summer had a close male friend and a close female friend, but they weren't allowed to be dating.
Here, the susceptibility to peer influence means the peer influence from the very two.
Yes, from those two friends. So not generalized from those specific friends. That's right. So we asked this about friend one and friend two, friend one and friend two. So it's not generalized. All right, enough yapping about the methods. Let me show you some results. So just a reminder, scan two subjects past summer. And compliance was really high. Each subject missed one checklist, total 98%, 56. Great. Two subjects, two female college undergrads, and these are their data. Or these are the findings. So first, you may be wondering, this seems cool, but are the data you're about to show me even reliable? You want to do these sort of you want to look at the data over time. How do we know that these sort of like neural indices are even going to be reliable in the first place? Well, I had the same concern, so I decided to pilot on myself, and I ran two 2-hour scan sessions where I basically showed every stimulus every time and some on myself, 19 hours apart. So I did it, I think, in the afternoon of Thursday and Friday morning. Great beating the magnets for a sixth of a 24-hour period. And I nominated two close friends of mine along the number line. Of course, I've known one for 20 years. I've known the other for 3 years, so it's not perfect, but it's as close as I could get. And I just looked to see. I analyzed the data by other to kind of gauge test, retest reliability. Here I'm going to show you stuff from a subset of the data because it's a little bit more. Enough time to go into the weeds on it, but I'm happy to talk offline, right? So if you just analyze the task data, like an hour's worth of those scans, 1 hour on day one, 1 hour on day two, and you get the signal that the task is accounting for in the variance of the brain data, the test retest reliability is 0.85, which is pretty good. If you break this down by other, so if I just model the task for other one and leave all the other unvariants modeled, or all the other variants unmodeled, and do the same for other two, it still beats sort of thresholds, or it kind of beats what else you see. It beats other metrics that you see with shorter scans. It was obviously not as high as 0.85, but this doesn't account for this variance. This variance is left unmodeled, right? And if you account for the variance of other two and modeling other one, then the reliability comes back. So this is the sort of like task variability that is just linked to other one. This is a task variability that's linked to other two. We're correlating that across sessions, right? So we just fit a model of the other one stimuli, other two. And this one I did do the, again, this is one of the weeds, but this is the voxel encoding model that kind of yields those atlases that I described way at the start of the talk. That was this, that was the framework I used for this. So it's pretty encouraging. All right, well, if the test retest reliability is so high, is there any even variability to model that? And the answer is yes. So what I did, let me orient you to this. What I did is I parcellated the data with the Schaefer 7 network 200 parcel atlas. And for each parcel, I went into one task or one other. So let's just start here. So I went into all 20 sessions for, say, the memory task, and I computed the contrast of other one compared to baseline. So I have 20 T-maps that were other one compared to baseline across all 20 sessions, right? And I took for each parcel, so we have a 20 by V matrix, V being the number of parcels, or V being the number of voxels in the parcel, and I computed the successive correlation between the voxels in each parcel to look at the stability across time. And I did this for all parcels, for both others, for both tasks, per subject. And you can see that there's are differences in the similarity between tasks, and a little bit between others, but not as much. So here we can kind of see that some of the ventral stream stuff in the memory is pretty stable in comparison to MPFC or DMPFC, or these cortical midline structures thought to be implicated in social cognition. And it's not that these guys are unstable; they're just they just show more fluctuations, which means there's some variance there that we can model. This is the same maps for subject 2. So you pointed out that there are these task differences, but I didn't see very striking differences between close others on that map. How do we know that these subjects are encoding close others distinctly? A great question. Well, they are. So if I take a univariate GLM of the other one minus other two contrast for all tasks, for all sessions, for all subjects, just put that in the second level GLM and control for subject, control for session, and control for task. Get this. And these are regions that care about other one, and these are regions that care about other two, just coarsely. So you see that there's this distinct encoding of two specific close others within the category of friends, right, of close friends. I'll say that one more time. I have an other one T-map for a given task and a given session for a given subject, and another 2 T-map. I difference them, and I took all those T-maps, so that's 20 sessions by 2 subjects by 2 tasks. And I modeled them and controlled for subject, controlled for task, and controlled for session.
Okay, we sure said it. I'm just. What is the map of other one based on, or any of that? Like, what makes it the other one unique? Like the trials that you ask about that versus implicit based on it? Like, what is it?
Yeah, so it's at the task level GLM. It's on the memory task is, are you answering this question about other one, the 4 or 5 seconds on screen, and then other 2, another 1, another 2.
Sometimes we thought that it's something related to reflecting.
Yeah, the other 1 or other 2. That's
the
right.
other one.
Yep.
Or the other 2.
Yep.
Anything you might be thinking about them, seeing them, hearing the voice in your mind.
I mean, anything related to the task. So what people experience during the task can be extremely heterogeneous, right? So you could replay a memory about someone while, I could see like, this is my like pet item. I like people say, I can't believe it was in the stimulus center, like goth night at the dance club. It's such a funny game. I might think about that like neither, I might replay a memory, right, while I'm thinking about that with a friend. Or I could be focusing more on how they would act if they were right at the dance club on God. Or I could be, zoning out or thinking about something else. So I really don't have control over what they're that fine control over what they're thinking. They do have to answer how well it describes each person, which I haven't analyzed those ratings. And then in the memory, they read back the memories.
So this is a contrast. So it says that some of these voxels clusters are more engaged.
Yep.
Than one than the other.
Hmm.
It doesn't say not engaged. So, for example, the ventral PSC region down there, you don't know. This shows just in this analysis that region doesn't care about both. It just happens to have activation. One of the
Sure.
the other. It doesn't mean that there's another categorical.
Would you say that that's still not a distinct signature, though?
It depends on what's driving it. If what's driving it is something trivial, no. If what's driving it is what you want it to be, which is something like an active mental representation, yes.
Mm-hmm.
But if you don't know, at least you will know, don't know this analysis, what is driving it, be something really profound.
Sure. Well, I'll have more data on that in a second. But yeah, that's a good point. But at this course, univariate level, there is differentiation among brain regions, right? So it's likely that I think it's unlikely that the core social cognitive regions in the brain just don't care about trying to represent other people's minds, but these regions care more about one other than the other in the context of this task, whatever they were truly doing during the task, right? And whatever sort of subcomponent of answering, does my friend like to live in a major city, right? Because it's kind of like probably a holistic or gestalt process that they're answering with, which you could break down further. Great. But do daily experiences track with these representations? So I took that same contrast, the same GLM I just told you, and I took these metrics from the EMA and I differenced them. I subtracted other two from other one, right? For time, cohesion, and susceptibility to peer influence. And I binned them so that they happen between scans. And for an EMA, for a response, I should stop saying EMA because ESM is data checklist, but for a response that came on scan day, that was lumped into the next set of scans. Because most of the scans came during business hours, and they likely spent time with their friends after. I don't know that for a fact, but I think that's a reasonable assumption to make. It's also one day among several that get averaged. So again, I've been the EMA, I've been the daily checklist data, so we're averaging in between scans, and checklists from scan day were used, but they go to the next scan, if that makes sense. So there's some temporal precedence here, right? I just want to point that out. We find that differences in time that you spend between other one and other two, fluctuations track with fluctuations, how distinctly you're encoding them, at least at the univariate level. And the same is true for cohesion, susceptibility to peer influence. And I'm not aware of another neuroscience study that has done another social neuroscience study that's done something quite like this. That shows at the level of, almost daily, from every couple days to every couple days over a 2-month period, that tracks how your lived experience, close others in your life, fluctuate your brain activity. Now, these are also, I ran these, it's the same GLM as before in the prior slide, except now I'm just adding a regressor. So still controlling for subject, task, and session effects, and I ran 3 separate GLMs. I'm glad that Michael made the comment that he made, you know, that this, you know, that could be kind of coarse. And you also just said just now that it's a single GLM. These are not controlling for the other. So we used RSA. I realize, Michael, this isn't going to quite get at your question, but I think the planned analyses will. We used RSA to look at how multivoxel patterns during this task encode these daily experiences. If you don't know about RSA, I'll just quickly say that it involves taking a dissimilarity matrix. You can think about it as a similarity matrix of multivoxel patterns for like a given contrast across every session. You have a square matrix, and you do the same for the daily checklist data corresponding to those sessions. And then we have a matrix for subject, for session, for task, and for other. So each similarity matrix was for one task, for one close other, for one subject. And then we just kind of compiled them into a big matrix, and we built the other matrices out the same way. And we put them all in the same regression model. And these are Bonferroni corrected here. So you can see that the time spent kind of has a little bit of fluctuations, right? Or that the neural representations are encoding information about time that you spend with them, albeit not as robust as cohesion or susceptibility to peer influence. And we see some of the usual suspects. We see cortical midline for items, but we see a bunch of and TPJ or so, but we see other regions that we don't ordinarily see in these social neuroscience studies that have a response with daily experience, which we didn't know before because we were just looking cross-sectionally. And with maybe like 10 minutes of task data as opposed to an hour.
Tell us about the chemical. Is that effect?
Yeah, so that's a great, that's a great question. It's not, I'm not controlling for brain or checklist at T-1. So it's just T. So it's just saying that for now, that like these guys, it's the daily checklist up to the scan, but not the day of the scan. So it happened before the event. Now you could have it could be the case that brain and checklist are contemporaneously associated across time and autocorrelated, and that's what's driving the effect. So having a lagged model is going to help in that aspect. I'm mindful of the time, so I am just going to sort of skip ahead for what's next, but it's to kind of use this encoding model framework. We can talk more about that for time. I will summarize and then see if folks have anything else to say, or I'd love to have discussion about it. I know I'm going over time a little bit, so sorry. But in sum, individuals encode close others distinctly based on a nuanced definition, rather than nuanced explanation. Encoding is reliable at very short timescales within 24 hours, but varies with daily experiences. And it's sort of linked to the usual suspect ROIs, regions that we know are implicated in social cognition, but also some that we don't know about. And that's all I have. Yeah, what's up?
Great talk, thank you. That's really cool stuff. One thing that kind of jumps out to me is that you're trying to get a lot of different sources at the same time, right?
Yeah.
So how subject one is representing others versus subject two, how representing one versus other two, how that's taking place in the context of the classes over time. So, you know, the big thing that jumped out to me, I thought was really cool, this fact that you see task maps that are dominant than representations of others. Right? Because before I saw the data, I was like, I could have seen it either way, right? You have representation of other one and that changes based on task. Yeah, versus what it seems like you're seeing, which is you have a task activation map that changes based on it. So then when you jumped ahead, those the final breakdown showed again, I believe you're saying considering all the tasks simultaneously there.
And yeah,
Yeah, is that like just about power? I think it's like it doesn't make sense to look at someone's mental representation if you're not looking at all.
yeah. I mean, this does control for tasks. I thought about running it. I mean, there's a billion ways that you can run it, and increasingly, I'm just thinking about let's just put everything in one model and let's put everything in one model and partition the variance accordingly. And really, that this, I think, like if you were to sit down without any of these maps or anything, and you were to write down, okay, where do you think the variance is coming from at this voxel at this time point? Okay, well, there is variance related to the subject, variance related to the type of task that you give them, and the time of day, and the stimuli, which we didn't account for in this, right? And lived experience factors and everything else, right? And so why not, if that's what you were going to say is in the variance, why not put it in the model if you can, right? Unless the model is so grossly misspecified or fits poorly. But this was sort of my first pass to it. Also, again, with only 2 subjects, I mean, what's nice is like the next 5 subjects are kind of like a confirmatory data set, or I could kind of like split it up in either way. I also wanted to limit the number of multiple comparisons off the bat that I made, even though this is quite aggressive multiple comparison direction on Romy.
I had the same question, actually, and you would do it in the same model, right? You would collapse across everything just like you did. You would just build an interaction for tasks.
Yeah,
You'd stay in the same model.
like in this guy, or
No, just in your original model where you said you collapsed across everything.
yeah, I didn't have an interaction between like task or subject or task or set.
I'm saying that's the way to get around it. You'd
Oh,
stay in
yeah,
the
yeah,
same Model, you would just include one.
Yeah,
interaction
that's right,
per
yeah.
task.
Yeah, so there's that. So there's high order interactions that are not accounted for, yeah, in this, but that's something that I hope to also run. And you can do that in this like encoding model framework as well. And what's nice is then you can visualize the R squared for each feature set, and you can have time or like, you can have like the EMA at each chunk, like and see how it interacts, the checklist at each chunk, and see how it interacts with task or ratings of confidence, like how well does this item describe the person, right? That was what they saw in the trade task at least. So you can go hog wild with this. There was a question back there.
Yeah. So I wanted to ask back to this neural similarity map. I'm just wanting to really check that I'm understanding correctly. But you were on the right one.
Oh, okay.
Neural similarity.
Yeah.
So I just want to check the neural similarity here is saying that between your 2 participants and these behaviors, these are the similarities, and the similarities specifically in daily behaviors, the daily behaviors being time cohesion, susceptibility of influence.
It's how much the time or cohesion or susceptibility to peer influence across the entire data set is encoded in multi-voxel patterns.
Yes. Yeah.
But it's controlling for subject, for task, so it's kind of generically saying for within these 2 close others, right? There's no difference between the close other that we're looking at. Yeah.
Yes.
Yeah.
And these 3 categories, time, cohesion, and susceptibility of influence, those are the daily behaviors.
That's right. Yeah. So if you think about it as like a time series, and this is time that you spent with close other 1 on day 1, 2, 3, 4, 5, and then you had scans in the middle. We just chunked them by the scans, right? And then assigned the days leading up to this, like the days in between scan 1 and 2. We sort of linked the average time spent with the friend for scan 2, and that's how they're linked here in the RSA.
Okay.
So there's been some criticism of the Galant encoding.
Yeah.
Well, I'm understating. And particularly people like Tim Rogers pointed out the inherent kind of implausibility of the results. And a number of people have argued that these kinds of encoding models are overpowerful. So they tend to be very promiscuous. I mean, if you look at, for example, Jeff Binder's encoding models looking at word features, you find almost the entire brain encodes every
Yeah.
every kind of latent feature that goes to define a concrete object. And the argument's been made that these things are almost impossible to interpret because of this. It's not that the data are not robust, but the interpretability is highly questionable. So Tim Rogers points out in one of the Gallant analyses, the main brain region seems to encode fear as the primary visual cortex. And this does not fit with a lot of other kinds of ways of analyzing similar sorts of data. So I'm just wondering, what do you see as the value of this encoding approach? When you've done this and you get these very complex maps, how are you going to interpret them?
Yeah, that's a great point, and I have a lot to say that I'll try to condense down quickly. But I'm so thrilled that you said that, and very timely because Tim gave a talk on that like last week. So like 5 days ago, he gave a talk on the data that he has, and he brought up some of these issues and stuff that he's doing to work around that. It's at my home institution. I think ultimately, what drew me to the encoding model is that it's this very highly flexible way to parse different sources of variance, right? But it doesn't mean that you can't use other framework. And I think that it's important to think about the fundamental assumptions that each model makes. And I agree with you that some of that, that when you're going to analyze the word embedding at the TR level, when there's a 10-second lag due to the HRF, and even if you use FIR functions, right, it's not really going to like magically make the problem go away. And I think that speaks to the importance of trying different frameworks and thinking critically about their assumptions and what they're giving you. There's no one magic framework, right? I'd love to do this with like EFIS patients, right? And analyze the data coming in also real time for like, spiky neurons, but I don't do that. So I think that it's going to be it's going to be taking the strengths of each of the different types of models, and there are various models that kind of get at this, right? Like RSA is one of them, decoding is another one, encoding models. So I think it's taking the strengths of each model and remembering to thinking about what their assumptions are and what purchase you have, sort of interpreting the gestalt. I will say, with fear and in V1, there is a gentleman at fMRI named Moreira, who's worked with, I think, Moreira. And they find that the visual system with fMRI seems to encode some information about emotion concepts, right? So there's multiple with a different modeling framework, and so there's multiple sources of evidence that show that may, I think that we have things that we think we know, and it's going to be hard to parse out when a new finding comes along. Is this just an artifact of the model, or is this something that we own?
Well, we do know that bilateral damage to the visual cortex doesn't stop people feeling fear. So it's unlikely to be necessary for the kind of core thing. I think whether or not.
Sure.
So this is the point that you don't know what is essentially epiphenomenal, what is actually telling you something fundamental about the mechanism.
Or how you define emotion and what components of emotion you think are tracking, right? Where in the brain. Definitely. Any other? I think I saw one more hand.
Yeah, it's kind of similar to the next, but I was just thinking about with the encoding, because you're looking at mental representations, but you need to kind of control how people are representing, so maybe ask them to answer the questions and represent it visually, like visualize your friend in 10 years' time, visualize how you do this. You don't know how each subject might be generating that representation, that might be sort of different scheme.
Totally. I mean, I think what's nice about these data is they're so dense that you could, analyze them and treat each subject sort of as its own data set, right? And interpret each of the maps individually like other folks have done. Or you can kind of think more deeply of what the shared variance across subjects means. I don't have a compelling answer for you. I wish I did, but I think other than these are the trade-offs with more kind of like rigid compared to more naturalistic experiments, right? The more rigid and sort of fine-grained and control that you exert, the sort of less it's going to generalize to what we're doing in everyday life, right? But the more you kind of let people be themselves, the more that variability comes out. And so,
But you need to ask them. What they think. So
say that again.
if you're not going to control it, you need to at least ask after. Like, what are you visualizing? What are you hearing their voice or whatever? So that you can try and control specific things or anticipate those regions being involved.
I could ask them after the fact. I don't know how reliable that assessment would be. Like, were you using mental image? I mean, maybe there's like a baseline could look to see if there's like how people kind of evoke imagery, right, and process that. Something, but
Yeah, I was just curious about like a key hypothesis you have related to study distinction between your post ranks or the key messages.
sure. Wait, sorry. Did
Yeah,
you-
just the hypothesis
Yeah.
that you have about this study, like your prediction.
Oh, yeah. I mean, I think there's going to be differences based on distance, but I don't know if that will be the strongest effect because they are relatively close. I think instead the story is more to see, like, to me, it's open-ended. Kind of have like you kind of have like a natural control condition by having two close others. But I will say, even looking at the data, like one subject spent more time with other two than other one during the study period. So I think for me, it's more of an open question.
Just for fun, what happened if you stop being a friend with that friend?
I can't wait. Well, so I didn't have time to go into it, but I will say for the daily checklist, we do have items. Did you have a fight with this person? And did you have a very strong negative or so fight is one question, then positive and a negative experience with each person, like borderline life-changing experience that you will take to the grave. You will remember to your dying day. And I think one subject had maybe one or two during the time, like one with each close other, and the other subject I don't think had any. And so, I don't know that they'll stop being friends with the person, but I would be curious to look at I don't know how much stock you can put into it, but I think it would be curious to see pre-post what the like presentations look like. I didn't ask them what happened. I should have. But yes, what happened? Assuming they would share it too. But yeah, I'm curious to see what would happen. Or like if you did this with a boyfriend and a girlfriend, they broke
Yeah,
up.
I was going to ask you, would it work for romantic partners as well?
I mean, I think you could run the study for sure. I don't know what, especially in there's so many open questions, right? What happens if you take a married couple that's lived with each other for 30 years, right? What happens if you compare an older person compared to a younger person, right? You know that as far as I know, I'm not super immersed in that literature, but there are ostensibly social cognitive deficits in older adults, right? So how does this vary as a function of someone who's 70 or 75?
Are there any questions online?
There are not any questions.
Right. Thank you.